In this class, we discuss Raman spectroscopy for the identification of iron Gall ink. We will see how to identify this ink on the main kind of supports, parchment and paper. As an example of parchment, we will work on these 16th century antiphonari, where the notes are made with iron Gall ink. As an example of a paper, we have this small document where on the top over here we have the date. So we have M, D, X, X, V and two lines, so should be 1527. This paper is a document written in Parma, Italy. Welcome to our Raman laboratory. Actually, this one is the lab where we have the spectroscopic methods. In this bench over here, that is a mobile, we can roll this one, is basically where we have the Raman spectrometers to do the on-site examinations. So basically we can put, like in this case, we have the pigment checker, we can put the pigment checker under the spectrometer and running the examination directly on site. Over here we have a different kind of Raman systems. Basically what is important in a Raman system is the wavelength of the laser, the excitation laser that we are using. So we have this spectrometer starting from 532, so it's a green laser. Then we have the red 600, then we have the infrared, the near infrared 785 with this probe on a fiber optics. Then we have 830 nanometers, so further into the infrared, and then we have 1064, that is the furthest in the infrared that we have. And why we have all this kind of different Raman systems? Because the problem is the fluorescence. So the fluorescence is a noise that covers the Raman signal, so what we need to discover. And so what happens is that when we move toward the infrared, so using these different wavelengths, laser, excitation laser, this fluorescence decrease, so we can see better the Raman peaks. On the other hand, when we move in this direction, so toward the infrared, also the Raman signal decrease its intensity. So basically the idea is that there could be one laser that is better for each application, depending on what material you need to detect and what is the binder, the support that are surrounding the material itself. For this application, the analysis of iron Gall ink on paper and on parchment, at least for the system that I have, the best one seems to be the 830 nanometer. So this one gives a good compromise because we have still a good Raman signal that we can detect, and the noise, the fluorescence is well decreased, so we can tell the actual Raman signal. So first of all, we do this little bit of an experiment. First, we test the iron Gall ink that is in our pigment checker over here. So we can acquire the spectrum of the iron Gall ink as a reference, and then we compare the other ones. So we will put the pigment checker over here. Okay, and we can start the acquisition. We are ready to acquire the Raman spectrum of the iron Gall ink standard that we have in the pigments checker. The pigments checker is over here, so this we are looking at the Raman system from the other side. So I'm in front of the computer. This is the software to run these spectrometers. Turn on the laser. First, we need to move the pigment checker so that it is actually on focus, the area with the standard. Okay, so you see here is we are seeing the iron Gall ink standard. So, and this spot, the red spot, is actually the Raman, the laser at 830 nanometer. So we go over here. So this is the swatch containing the ink. So it's over here. We can start the actual acquisition. And we start to see the signal of the ink. Okay, so basically we see a lot of fluorescence, and then we have this peak over here, and this other one, and another one at 600, very smaller over here. So these are this is the kind of signal that we get with Raman spectroscopy for this ink. So basically the signal is very weak. We save this one and we proceed to acquire the spectra of the parchment and the iron Gall ink on the parchment. This is the antiphonary parchment, 16th century. It goes under the spectrometer over here, and we want to analyze, like for example, this spot over here that is iron Gall ink. Okay, and we can go to the console for the actual measurement. We do the same for the document on paper dated 1527, Parma, Italy. So we measure both the paper and the ink, and so we did the same also for the parchment. We measured the parchment and the ink on the parchment. This is important because we want to be able to distinguish what peaks belongs to the support and what peaks belongs to the actual ink. Okay, ready to measure. Turn on the laser, and in 5 seconds we should start to see a spectrum. There it is. So this is the spectrum of the paper. We did acquire the Raman spectra of this antiphonary parchment. So we acquired the spectrum of the parchment and the spectrum of the iron Gall ink over here. Then we edited the spectra, so we did a bit of a baseline adjustment because these spectra are very affected by the fluorescence, and we represent all the spectra over here. Okay, so we start with the blue one on the bottom, this one. So this one, as in the legend, is the pigment checker iron Gall ink reference. So this is a typical spectrum of an iron Gall ink. So where we see basically the main feature to recognize it are these two peaks over here, this one and this one. So I put also this line to follow and compare to the other spectra. Then there is another peak over here at about 600. Now we look at the spectrum of the parchment alone. So this spectrum, we start from here. So we have this big peak that is the primary amide. So basically these are the spectra, these peaks comes from the collagen, since basically the parchment primarily is made of collagen. So we have this big peak over here, amide, primary amide. Then we have this other peak over here, the tertiary amide, and the secondary amide are in this region. Then we have these other relatively strong peaks here from the skeletal carbon-carbon bond that are in the collagen structure. And then we have also this peak to the proline skeletal also structure of the collagen in the parchment. Okay, so this basically is the shape, the Raman spectrum of the parchment itself, just that. Then on top, eventually, we have the spectrum of the iron Gall ink on the parchment. So we see strong peak over here. This one is the peak of the ink. So is this one, the same over here, is actually stronger than the one we have in the pigment checker. Then we have this other peak over here on the second one, the strongest one of the ink. On the back, there are the spectra of this document on paper. In the blue, on the bottom, we have the spectrum of the reference iron Gall ink. And so we see the main peaks over here, one and the other one over here. Then this one on top is the spectrum of the paper, where the main features are this strong peak of the cellulose in the paper. And then basically the black spectrum is that of the iron Gall ink on the paper. So we see clearly the two peaks of the iron Gall ink. So basically now in this small course we discussed what we can expect in the measurement, the Raman spectroscopy measurement of the iron Gall ink on the paper and on the parchment. Thank you for watching.